This lecture is about protein sorting. So, where do proteins go after translation occurs? There's three main categories that we can think about: cytoplasm, the organelles, or the endomembrane system. So, on this page, you can see at the top, we're showing translation process where there's a messenger RNA coming together with ribosomes and the new polypeptide is being formed. So it turns out there's two types of protein sorting. One type is shown on the left. It's called the co-translational import, which means that the proteins are going to be imported into the ER membrane before translation is completely finished. The picture on the panel on the right shows that the ribosomes are actually going to finish translation in the cytoplasm, and then the protein will be transported into the nucleus, the mitochondria, the chloroplast, or the peroxisome. And so that's called the post-translational import. Most of this lecture will be dealing with the co-translational import. So what determines the final location of each protein? Turns out to be a amino acid sequence at the front of the protein on the amino side of the protein, and that localization signal will have a specific sequence depending on where that protein is supposed to go. If there's no localization signal or no tag, then the protein will stay in the cytoplasm. But all the other proteins that need to go someplace will then have a signal sequence. So my analogy to this is a tugboat guiding a ship to a dock. The tugboat is going to be our special molecule called the SRP. The ribosome is the huge ship, and the dock that we're trying to get to is the ER. So I'm really talking about co-translational import. How do we get a protein to the ER membrane? So before I show you the pictures, there's a few definitions. There's something called the signal peptide. That is a signal sequence, about 15 to 30 hydrophobic amino acids at the front end of the protein. And there's a particle that binds to that called the signal recognition particle. It binds to the signal peptide, and it actually blocks the ribosomes from doing any more translation, and then it brings the ribosome to the dock. And the dock is actually a SRP receptor protein that is on the ER. So this idea of co-translational import was originally called the signal hypothesis by Blobel and Sabatini, who later then won the Nobel Prize in 1999. So the idea is there's a free ribosome in the cytoplasm that starts to do translation and makes about 15 to 30 amino acids of this protein. If this 15 to 30 amino acids happens to be the hydrophobic ER signal peptide, then a signal recognition particle, or the SRP, will come and bind to the signal peptide. And the SRP will then bring the entire protein and ribosomes to the ER membrane. And at the ER membrane, the SRP will bind to the SRP receptor, and the ribosome will basically rest there. And there happens to be a pore on the ER membrane channel. The channel is called the translocon. So when the SRP leaves its receptor, what happens then is translation is allowed to continue, and the protein that is now elongating has no place to go except to fall through that hole, that ER membrane channel. So, but as it falls, the front end, that signal peptide, because it was hydrophobic, it will bind to the phospholipid bilayer. And then that forces the rest of the protein to continue to fall through the ER channel. After translation is completely finished, then of course the ribosome will disassemble and the protein will fall through the channel, but still hanging on by that ER signal peptide. So there happens to be a enzyme on the membrane called the signal peptidase, and it will cut off that signal peptide, and so then the protein will then fall through and be completely into the ER. So this is another view from a different textbook. So in this case, the red is the ER signal sequence and the green is the polypeptide. And you can see the blue is the translocation channel. And so what we're trying to do is we're trying to make a protein that comes to the ER and then falls through and stays in the ER lumen for a little while. And then remember the ER, then that protein will then be glycosylated, put into a membrane vesicle, sent to the Golgi to be glycosylated, and then delivered to wherever it's supposed to